Okay, so this is Muscle Week, and if we were here live, you'd be happy because this is a short lab for me in terms of my instruction. I simply go over the microscopy of cardiac muscle, skeletal muscle, and smooth muscle and help you to find the difference when you look under a microscope. So they'll actually give you a microscopic slide and they're going to ask you a few different questions about it. But let's start off with cardiac muscle. All right, you see the real micrograph here and the cartoon. One of the things that identifies cardiac muscle is that the fibers are not parallel. They actually converge on each other. So where you see these convergence or forking, depending on which way you're looking, if you're going this way, it forks out into two. If you're going this way, it converges. All right. Another telltale sign of cardiac muscle is these very, very dark lines that seem to divide the muscle up into strange little cells that are not always the same shape. You look at this one, it looks like different plots in a housing development or something like that. Down here you got more elongated ones. But what is dividing each one of these are something called intercalated discs. Intercalated discs. These are your nuclei. All right. So what's an intercalated disc? Let me see if I can show you a little closer. Here you see the intercalated disc pointed out in the video, and I'm going to try to go a little closer. All right. You also will have this PowerPoint yourself. But these are the intercalated disc, and again, this would give away cardiac muscle. You will see striations in cardiac muscle. All right, give you an idea of what that intercalated disc. Remember when we talked about tight junctions and desmosomes? It's how cells are stapled together. Then there are connexons, type of proteins, linker proteins that are there for gap junctions. So what happens since the cardiac muscle is under extreme stress? You need to hold these cells together with a lot of desmosomes, a lot of tight junctions. And that's what an intercalated disc is. This figure is from your book that shows the intercalated disc. Again, you can see the zipper effect because you have these linker proteins that are holding it together. They are protruding from one side of the muscle or the muscle fiber or cell to that cell, and there's just tons of them along with gap junctions. All of those gap junctions, all of those desmosomes, that is what makes up an intercalated disc. Okay. Again, just showing a bunch of intercalated discs, the striations, the nucleus. So when it comes to cardiac muscle, no matter what kind of stain you use, blue stain, purple, erythine, red, the intercalated discs, the fact that these are not perfectly parallel, it's harder to see that there, but the intercalated discs definitely give that away right there. All right, skeletal muscle. Skeletal muscle is perhaps the most voluminous muscle that's in your body because it's attached to all of your bones, and what gives it away is that these fibers are absolutely all parallel. Okay, and you see the many striations. The nuclei are not as large. You might be thinking, hey, there's an intercalated disc. Okay, that might look like one. But we don't see the fibers going off in different directions, nor do we see the amount of nuclei that are much rounder. Since skeletal muscle is under stretch, these nuclei have a tendency to be elongated. All right, so that's what kind of separates. Look at the elongation of the nucleus, and then these are the many striations due to the crossing over of actin and myosin fibers. And we've talked about this in our book. We talked about the H zone, the A band, the I bands. In between each I band is the functional contractile unit of the muscle. I don't think you'll be shown this, but I am showing it for just in case you are. Now, the thing about skeletal muscles that you have to understand is that you can look at it from the surface. Okay, so imagine cutting this muscle like this and then pulling it back and looking down on it. Okay, it's as if this muscle fiber is rounded right here. Okay, and as a result, you have a lot of other bundle fibers right here. Okay, and those bundle fibers, this is what they did. They cut this, and now we're looking at these muscle fibers right here. So this is still skeletal muscle. Okay, let me erase that or forever be. Okay, so I hope you got that distinction. Skeletal muscle. Well, what's all in between the muscle fibers right here? That is just what we call extracellular space. Could be packed with some connective tissue, some adipose tissue. And then another way of looking at these muscle fibers right here. Here's the surface of the muscle. That was where you see the striations. Cut the muscle in half and you see all the different fascicles that make up skeletal muscle. And again, this is the cross-section of the muscle as opposed to looking at it from the surface. Okay, so imagine these fibers are rounded, okay, and then you cut them in half and then they have their individual fascicles. You know, if you're ever having a bad day and you just want to feel better, just say the word fascicle. It just kind of brightens up the day. All right, let's talk about smooth muscle, and I'll be out of there pretty soon. Speaking of which, I'm doing this from my high school room. I've got a much better board here than what's over at the lab. I don't know why I didn't think about it. But anyway, the interesting thing about smooth muscle is that smooth muscle basically runs in two different directions. Okay, if you have a hollow organ like intestine, all right, in the intestine you want to have muscle that wraps around and then also goes the entire length. Okay, so depending on which direction the muscle is going, we call it longitudinal because it's going the length, and then you have what we call transverse or circular smooth muscle. Okay, let's see if we get a good picture of that somewhere. All right, so here is the longitudinal. It's running this way, and then this transverse is coming at you. Okay, so this muscle is running two different directions. Okay, so what Eric could do is say, what kind of muscle is this? Obviously that is smooth muscle. All right, but he might say specifically number 2, that is what kind of muscle? And that would be longitudinal. And then he'll look at this area and say, okay, this muscle is, and you would say transverse. Okay, so transverse cut, that's kind of basically taking this and then pulling it back so you can look at it. That's what that would be right there. All right. So for the most part then, you got your cardiac muscle with its non-linear stuff right there, intercalated discs made up of desmosomes and gap junctions. Okay. Then you have your skeletal muscle, which can be looked at the surface, or cut this, turn it, look down, and you're looking at a transverse cut, but we don't call it transverse skeletal muscle. It's just skeletal muscle. We see the fibers from a transverse cut. Okay. But we do call the transverse cut of smooth muscle transverse cut smooth muscle, okay? Or longitudinally cut smooth muscle, okay? Those are the two. Longitudinal, transverse. Okay, and that will be the end of that. Yeah, I'll turn it off somewhere. All right, so in your content folder, I was able to put the torso models that are labeled, along with the appendicular arms and legs, or no, the legs, yeah. So that is what you are doing this week. You're looking at all those different muscles, and look at those pages in the lab manual where you have those charts that are already filled out for you. Remember the microscope way back when, where we had the part and we had the function, all right? And then we had the tissues, where we had the type of tissue, we had epithelial and connective tissue, and then where it was located. So you had to memorize that chart, all right? Then you did the skeletal system, the axial skeleton, and there wasn't any chart there to really know. But there was a nice little chart or boxes that show the articulations for the next week, and that's what you did last week, articulations. And so this week is about muscles, knowing the type of muscles, making sure you know for the selected muscles that are in the lab book, you need to know what is the prime mover and what is the antagonist. And a good example of that is the bicep is the prime mover of the arm, and the tricep is the antagonist that returns it back to where the bicep took it from, all right? And I think there's, I don't remember how many, but there's a little box there that says, you know, it has the antagonistic muscle and whatnot. So that is what you'll be doing, and I think I'll help you out when it comes to the day of the lab, in which I can tell you how to fill out. Maybe I might do it now. Let me see how many minutes are left before the stupid video cuts off. Yeah, we're 13 minutes. Let me see if I have my lab book here. Just wait there for me. Okay, yeah, so I don't have my lab book with me, so I can't do that. I'll have to do it when we do our labs, or I might make another little video and ooh, I didn't know that made it bigger. I could have done that. There you go. Alright, I'm trying to get out of this thing. There it is. Alright. I have to send this over there so I can pick it up. Alright, so that was all. Kind of short lab because most of the stuff is in your content page that you're going to memorize. And we'll just leave it at that. So we'll see you. Bye-bye.